This is GCSE Biology Required Practical about how light intensity affects the rate of photosynthesis in a pondweed. Photosynthesis is a process through which green plants are able to manufacture glucose using water and carbon dioxide in the presence of sunlight. Oxygen is produced as a byproduct. The factors that affect the rate of photosynthesis are 1. Light intensity, 2. The concentration of carbon dioxide, and 3. Temperature. So our independent variable is the source of light. That's what we are changing, the lamp. Our constant variable is temperature and also the concentration of carbon dioxide. You will need to calculate the light intensity by measuring the number of bubbles produced by plant per minute. You will also need to get a graphical representation of the number of bubbles produced per minute against the distance of the pondweed from the light source. So place a piece of pondweed, Elaudia in this case, into a beaker containing sodium hydrogen carbonate. This is to encourage the pondweed to photosynthesize. Place in a glass funnel over the plant in the beaker and start the light 10 centimeters from the plant. Record the number of bubbles observed per minute, and then repeat these steps for different distances of 20, 30, and 40, and 50. So we've turned out the light of the lab, and so the only light that we'll have on is our lamp, which is our source of light. We've turned it on now, and we start with counting the number of bubbles produced per minute when the plant is 10 centimeters from a light source. Sometimes the bubbles are hard to spot, but if you look closely, you can actually find them. We then move our plant to 20 centimeters from our light source, and we do the same. We count the number of bubbles produced per minute. And we do the same. We go to 30 and repeat the procedure. Once you've counted the number of bubbles per minute, you go to 40 and 50. How can we improve this experiment? We can do this in 2 ways. First, we can repeat the experiment twice for each distance to get a better result. And secondly, we can place a water tank between the light source and the plant to avoid heating, because what we don't want is temperature to be an independent variable. We want a constant temperature, and so by doing that, we then keep the temperature constant. So you'll find that the number of bubbles that you get per minute decrease as you get away from the light source. So the closer you are to the light source, the more the bubbles, and the further away you are from the light source. You can then put these results onto a table, and you can then translate this table into a graph. You can put the number of bubbles on the y-axis, and you can put the distance of the plant from the light source on x-axis. This experiment is part of a series of GCSE required practicals. To find out more, you can visit our website at tiny.cc/HVS required practicals.